From the heart of preLights, the preprint highlighting service run by early career researchers and supported by the Company of Biologists, this is spotLights. Hello, and welcome to spotLights, the preLights podcast that highlights the stories behind the most exciting biological preprints, putting the spotlight on the early career research who spearheaded the preprinted work. My name is Matthew, joined by Ethan, and will be your host today. In this episode, we're joined by Daniel and Oskar. Today, we'll discuss their preprint TAC1 operates at the primary cilium in non-canonical TGF-beta/BMP signaling to control heart development. Welcome both to you, Oskar and Daniel. Before we get into the first question, let's start with you both introducing yourselves to us. What are your backgrounds? Oskar, should we start with you?
Yeah, we can start with me. So my background is in mostly molecular biology with a slight twist of medical sciences and health education involved. Most of my studies involve cellular interaction and signaling. So the proponents that I study will almost always relate back to some kind of cellular signaling process. Which is also quite clear in the preprint that I think we're going to be discussing today. I'm a PhD student in the laboratory of CERN for Christensen, and we focus on this tiny organelle called the primary cilium, which is a bit like a cellular antennae. And right now I am studying both heart and brain human development during embryogenesis.
Very nice. Okay. Daniel?
Yeah, so I'm Daniel. So my background is also mainly medical sciences and also the research related to that. And so initially when I first started getting into research, I was involved in a nanotoxicology study at the University of Edinburgh. Then eventually when I progressed through that, I ended up in the cilia field, which is obviously what Oscar's just been talking about just now. I ended up doing my PhD in Copenhagen in the labs of Lars Arne Larsen, who works quite well and quite a lot with Oscar's boss, Søren Skov Christensen. So that's how the two of us have actually combined together and to sort of produce this preprint that we'll be talking about, and then kind of how I got into the whole field, really.
Mm-hmm. Right. So you started, or you moved into cilia related work, and
Yeah,
Oskar, you were looking at signaling mechanisms. Did you both have any experience in developmental biology before then?
Well, that's an interesting question. So not particularly, I would say. So to me, my start was really the cellular signaling aspect.
Okay.
But because cellular signaling, particularly in cilia, have a tendency to either relate to some kind of syndromic defect or the sorts of progenesis or origin of the defect, you either end up studying something that's related to a human disease later on in life or a developmental defect.
Okay.
And due to the signaling pathways that we were interested in at the time, which were calcium signaling and something called transforming growth factor beta superfamily signaling, they both relate heavily to developmental processes, more so than at least particularly some of the later life diseases involving cilia. So in that sense, the signaling kind of developed into the developmental sciences field more so than the other way around. So for us, it was more of a necessity to go into the field and really explore what was going on.
And for me, it was actually kind of the opposite side, where I was initially doing research on a certain syndrome that was related to cilia called primary ciliary dyskinesia, which I was doing my master's research project on in Leicester. And so taking it from that perspective, I then went to Copenhagen to kind of do a bit of somewhat disease modeling when it came to the primary cilia, when it comes to heart development. And then that's kind of how I got into the more developmental side of things by taking kind of the opposite route from Oskar coming up from the other side.
Okay, cool.
Actually wondering how both of you get interested in the first place. Now that you're doing your PhD and transition to postdoc, where did you start?
it's a big question, isn't it? It goes quite a long way back. But for me, I think science just always has been a major interest for me. I'm obviously, you might be able to tell, I'm from Scotland, but I'm actually from a little village outside Edinburgh called Roslin, which is where they cloned the Dolly the sheep in 1996. So that was obviously a big part of like the education and the sort of history of the village that I was living in. And so with that, I got interested in genetics quite a lot and kind of decided I wanted to be a doctor, initially a medical doctor. Then I'd done some, the usual, you go to like shadow doctors, things like that, and realized I actually preferred the more behind the scenes aspect of the science, which resulted in me doing my degree in medical sciences, then the master's, and then the PhD in the end. So my kind of interest from science really stemmed from quite a young age, and I suppose don't think there would have been any other career that I would have been interested in, given the background.
Yeah, so I think mine is somewhat similar, but it's more so. My dad is into structural biology, and he always asks me these random questions like, "Oskar, you need a favorite amino acid," and I thought that was completely stupid. I still do think that, I should add.
And then,
but when you keep probing the lion's den, you put a stick in there, eventually something comes out of it. And I remember I had this. I went to a really bad school. I transferred, and then there was this guy that asked me a question regarding how do we even think, and I was like, I don't know. Don't need to think so hard about it. But then you do think hard about it. Horrible question. And then I got really into neuroscience, and I thought I was going to study neurology as a doctor, so you become more into the neurosurgical part. But I kind of didn't have the interest of pursuing patient relations, because staying on the sidelines is just more my thing, I think. So to that end, I ended up being very interested in the underlying dynamics. And then I encountered the signaling aspects. And in neurology, you have all the, the different signaling pathways, just with axon terminal synapses and so on. But this was just every cell that did it. I thought that was really fascinating, because you don't really encounter that very many places where everything just constantly signals back and forth. It would be a bit like if every single object in a room was an antennae. Seems a bit ridiculous, right? So I thought that was really, really fascinating. That's how I kind of progressed through the field in terms of interest.
What is your favorite amino acid?
What is my favorite? My favorite one at this point is tryptophan, because it's the biggest one.
Oh,
follow
mine's
the bigger the better
one. Mine's the smallest one. I like
Do you like
glycine.
glycine?
Yeah, exactly.
Did that answer get your dad's approval, Oskar?
No, he was very disappointed, because it's like, it's just big and bulky. It doesn't do anything. He was very disappointed that I didn't choose anything with a charge. I'm a very non-polarizing character, so maybe that's why.
That's perfect. That's perfect. So, what did you find most challenging in your PhD career?
Right. I think for me, because I had to make the move from the UK to Denmark, which it's not a massive difference, but I moved during the COVID pandemic in 2020. So actually getting used to a new country, the new sort of paperwork, the bureaucracy, and how to interact with people, which Oskar probably say I still don't quite understand. Yes, it was a little bit tricky, kind of more the social side for the PhD for me for quite a long time, just because of that. But then, of course, you end up meeting people like Oskar. He was very good to me. I'm sure he'll nod in agreement, or you'll just be thinking, what an idiot. But,
no. So I think I imagine moving to Denmark during Corona must have been hard in itself. But Danish people are like the most closeted people I think I've ever met. Like, you go to like the States, everyone's like, "Oh my God, welcome, hello, how are you doing?" You go other countries, they're very warm and welcoming. Denmark, you look at each other like, "Please do not talk to me."
Just a little bit of disdain. You've got to kind of get the look quite right.
I think they call
You've
it
got
the
to practice
side eye
it.
sometimes, right?
Yeah,
Where you look like, leave me alone.
But no, these things just take time in the PhD as well. Even like, because normally when you're doing like a PhD, you'll have like a big group of you, maybe in the sort of the intake of that time. But of course, because it was COVID, we couldn't really do anything like that until. So I started in October 2020, and then we started to do more things openly in Denmark. I think it was about February, March 2022, because there was a few other things going on in the middle.
The rings
and then I made the perfect decision at that point to go on secondment to Spain, where they still had COVID regulations. So I was following the rules at the time. Just, I don't know, maybe I just hated myself at the time for some reason. So.
Yeah, but you did have the nice support of the program you were in with. It was a very social program.
That is the other thing, which is actually probably a really big positive for the PhD program I was on, because I was on a Marie Curie scholarship program. And so it was called the Skills Consortium. So it stands for Studying Ciliary Signaling and Development of Disease. And there's 14 of us in total, all across Europe. So there was some of us that were in Denmark. I think there was 4 of us in Denmark. Couple of people in France, Spain, the UK, Germany. I think that's it. And the Netherlands as well. So we're all kind of PhD students working on a very similar project, all looking at the cilium, but in different labs all over Europe. And so we may not be able to have seen each other regularly in person, especially at the beginning, but we did have this whole network together, and by the end, we're all quite close. And so it's quite good to have, that was a really good aspect, because you've actually made friends and made connections that are going to be useful for your career in the future as well. And also just social life, you get to go and, if you're just in a random country, so they'll all come meet you. Quite easy to do.
I had pretty much the same struggle as you, because I'm also studying abroad now. I'm in Germany, and I moved during COVID as well. And I had not only COVID to deal with, but Brexit as well, of course.
Yeah, of course I had that as well. So
yeah, so had all the bureaucracy with that as well. So yeah, I identify with that a bit. Yeah.
Crazy time.
Well, hopefully it'll get easier.
Oh yeah, yeah. I've settled in now. I'm 3 years. Yeah. Much better.
Lovely country, Germany. Once you're finally there, right? I mean, good place, good people.
Yeah, definitely, absolutely. Really, really friendly people. Absolutely will do anything to help you settle in.
Mm-hmm.
Good beer as well.
I love the
food. Yeah, good beer. Mentioning October as well, it's a good time to be in Germany right now.
I'm going there for an EMBO meeting end of the month, and I can't wait for the pork knuckles.
Pork.
Is that Ethan?
Heidelberg.
Okay. Yeah.
Yeah. I can't wait. Maybe we'll meet up there. So, if I'm, wherever you are, we'll figure it out later.
We're on the way.
Yeah. Awesome. Cool. Oskar, did you talk about your?
No, so I didn't.
Yes.
I think one of the greatest challenges that I've encountered really is the specification, because you only have a set amount of time, but for at least what, and I'm sure Daniel will agree to that. I'm thinking some of you guys will study something where the broadness of the matter is really, really enhanced by the amount of different avenues that you can go down. So for cellular signaling, it's just involved in so many processes, and we can't really figure out how interconnected all of these different processes actually are. And that presents a lot of challenges, because you end up doing a lot of nonsensical work to kind of figure that out. And when you then have a limited amount of time to actually complete it, including having to do animal studies in zebrafish, cell work, take care of students, do teaching, all these other things, I really think the matter of time, but also self-control to kind of say, this is the one path I'm going down. It's really hard to manage when you're this early. Well, I'm just finished first year. So when you're this early on, it's hard to really set yourself on that path and limit yourself. And I think that's a continuous challenge that I'm going to encounter.
I'd agree totally with that. When it came to the project that Oskar and I we worked on 2 projects together, I believe, but the one that's been out, well, they're both.
I think technically we worked on 3 projects together. It's just one of them is a bit far-fetched.
Oh yeah, it's quite complicated, but we won't get into the full details because it's a bit logistically boring. But we were working on one of the preprints that we've got that was out, and it was kind of more of a decision of what is actually the interesting thing that's going on here. And of course, technically, it's all interesting. But given that you've got, say, 2 or 3 months to get these experiments done to try and get these papers out, you've got to somewhat be lucky in your decision making as well to be like, which one is going to be the best? What's going to give you the best data and what's actually going to help you with the story of the project? Because a lot of the experiments that you end up doing don't really add that much sometimes, depending on how it goes. I mean, maybe a nice example of that Oskar and I, because we kind of first met properly while working on a different preprint that's to do with a gene called ADCY6. Now, initially, the idea was we wanted to try and see if this protein from this gene is found within the cilia. And so the plan was to take sections from like mice at certain developmental stages and then stain them, look at the hearts and see, do we see this protein there or not? Now, getting that to work, well, it didn't work, but trying to get it to work, I think took a couple of months.
It took a couple of months, and the annoying aspect was really that you get these kind of false positives.
Mm-hmm.
So immunohistochemistry is very susceptible to any kind of particulates that end up on the tissue sections, particularly on old tissue sections. And because cilia are these kind of small, elongated structures, anything looks a bit like a cilium if you're not very careful. So we kind of got these results that were like, oh, is it there? Is it not there? We should continue going to figure it out. But, at the same time, we had the feeling it wasn't there because we've done this experiment like a billion times at this point. But you get the sense that we're progressing without actually progressing. And that just took a lot of time.
And then you get to the point where you just have to make a decision of, is this actually worthwhile? And at this point, it was no. So we moved on to the cells instead, which Oskar is more of an expert at than me. And so he continued with that side of the research, and we turned out to actually get a good result in the end. But it just had to be that sort of fork in the road of what do you do now?
So you mentioned Oscar's more into the cell culture side of the project. I guess you're more into the zebrafish side of this. I noticed there were, is it right, 4 equally contributing authors to this preprint?
Yes.
How you shared the load between you all?
Yeah, so it started off with Jaden, who kind of started the whole project, how many years ago it was. And then I jumped in, I would say maybe a little bit into the project. And so myself and Jaden were doing more the zebrafish research side of things. Then we had Oskar and Yasmin. Yasmin is another PhD student that started before Oskar, and Oskar's coming in to try and help finish it all off and get some new data for it now. They were both doing all the cell stuff. And so it required a lot of communication between our lab groups to actually figure out what on earth's going on with these genes and proteins.
And also actually something for a while, I think we can admit fully that the communication between the lab groups was probably holding the project a bit back. I would reckon you'd agree with that, Daniel, wouldn't you?
Yeah, it got to a point where we thought we'd finished the zebrafish data and we needed to get the cell data. And of course, people are doing lots of different things at the same time. So it wasn't really moving forward. Then Oskar and I kind of came together, and we're not going to take the credit for it, but we will. That we kind of allowed the projects to develop a bit further. We also came and done this, the cell data, which then helped us a lot with the actual zebrafish data, because although we thought it was finished, it wasn't, because he found some interesting stuff. So that allowed us to actually really focus in and try and get it all done as quick as we could, which turned out to be a year, but we got there in the end.
I think one of the primary key examples of the lack of communication was for a long time we had all the interesting zebrafish dates and we had all the interesting cell dates, but the actual connection between the two is a bit loose, because we can very easily reason that the proteins of interest, which were TAC1 and TAC2, for example, they would be in the cells, but we never documented their ciliary presence in zebrafish. at any time point doing this. And I think it was almost because the assumption was that it was just a given that it would be there, which, it's a fair enough assumption if every data supported each other perfectly. But our experience was also that there were a few other holes that needed to be patched up to really get there. So we decided to speed up the process by making a pipeline between ourselves, where we could very quickly formulate either a whole mount protocol or tissue sections to then immediately go investigate ciliary type 1 or type 2 in the zebrafish hearts, which is a mightily problematic thing due to the lack of antibodies, but at least it sped up the process of actually trying to connect the two sides of the stories. Because it was a bit of a bipartisan story. I think you would agree with this as well.
Yeah. And I think that's kind of how Oskar and I kind of came together as a team at that point. I mean, we'd worked a little bit with this other ADCY6 protein, but then it was this TAC1, TAC2 stainings and further cell data, which kind of cemented, let's call it the working relationship between the two of us. And we actually, I think we'd say we got a little bit lucky when it came to the zebrafish immunohistochemistry, because zebrafish and immunofluorescence and antibodies are notoriously difficult to get working, to get right, and to actually analyze. But luckily we had a lot of antibodies for this TAC1 protein, and we had a lot of zebrafish samples to use. So I think we must have taken like 6 different combinations of antibodies with 20 slides or more than that. And a lot of them just
counted them, and we had a lot of different combinations. It's not only that we had a lot of combinations; we had a lot of different preparations for everything. Yeah. So I think we in a few months we must have done well over 100 tissue sections of data, just plowing through everything.
And then I think there was just one time we were at the microscope with all these different combinations of stainings, etc. Most of them hadn't worked, but there was just this one sample we picked out. Turns out it did work for that one slide. And luckily we were quite early on in the microscopy session, because if we'd gone any further and everyone was just, no, we might not have even reached it. And so we've got a little bit lucky with that. But it just shows you sometimes you just need to have something like that happen.
I think it's really important we got it early, because you might imagine the depression of sitting in this dark microscope room. Yeah. I'm sure you can relate. Sitting in that depressing microscope room, you have 100 sections or whatever we had. I think we had 30 that we were investigating in that section. And we had 5 earlier, and it takes a while to look through them, because you have to look the mutants, the wild types, you have to look through every single heart. And you just get more and more down every single time. It's like it's dark, you're tired, it's humid, and this microscope is just making this constant bustling sound. It's like white noise. That helps people fall asleep. It's not supposed to go in the microscope room.
Yes. And I have a memory of this being on a Sunday as well.
I think it might have been. I think we were very desperate at the time. Yeah. So that was a very long day.
Yeah. So would you say that's the highlight of this project, or
I think I think in terms of a singular moment for that project, I think that's the one I look back at the fondest, because it a lot of the other times you have an inkling that something will work. Okay, we've kind of optimized further and further. We're getting somewhere, but that one we had nothing that worked, and then works. I think that was amazing.
I would say the same. I think also at that time period, there wasn't actually a lot of cilia stainings in zebrafish. When you look at the actual literature, there's some, but it's not an awful lot, and not doing the technique that we were doing. And so the fact that we actually managed to get it working ourselves was quite a joyous moment. I think we probably went to the pub straight after.
Yeah, so there's this lovely Irish pub in Copenhagen. There might have been some rugby on it the day as well. So it's really good timing.
I don't know if I could get any more British if I tried.
The Irish would be very impressed hearing that.
They probably would be, but I mean, going to a different country, going to a local bar. No. Irish bar. It's next to the English bar. It's also next to the Scottish bar. So probably done a little pub crawl.
I can inform they make lovely fish and chips. So we definitely indulge in some British traditions in that moment.
Yeah, I actually live next door to an Irish pub as well, so I'm guilty of that as well.
So we're doing well.
Yeah. And I was wondering, since this preprint was posted on bioRxiv a number of months ago, give us an update now. What are you both doing now? And is that the end of that project? Where's the project lie now?
I'm sure Oskar will probably say, well, the
Oh, yeah.
project ever end. I'm not quite sure it ever will.
So this project is well. So first and foremost, we need to get everything sorted so we can actually finally get the preprint published. So until that is completely out of the way, it's a bit on the back burner to actually continue everything.
Yeah,
there are two ways the project would be interesting to continue. One is basically a zebrafish study functionally, because one thing that was very notable was this skeletal phenotype that Daniel did a really good job figuring out. And because that also could indicate that there are other phenotypes that we haven't actually fully discovered, because for a long time we didn't know that this was there. We just kind of assumed that the heart phenotype was the end of the story, and that would be what we focused on. But then Daniel, I don't know how you got there, but he got the idea to get into that. And it really showed that there must be other aspects of disease that could be very interesting to model through the system. One thing that's very notable in congenital heart disease, which is not quite very apparent in a lot of functional studies, is that there is often a brain phenotype linked to heart phenotypes. And when we have other syndromic defects, it's very likely that there might be a connection there that we just never actually got the opportunity to investigate. Another aspect would also be to delineate the cardiac structural stuff, which I think Daniel will be better at. But it's very notable that the cardiac chambers exhibited less trabeculation, because that's not really something that we can corroborate with the cell data necessarily, but it's something that functionally is quite interesting and would give a human phenotype. So maybe you want to speak first on that before we go into the cell signaling stuff that's very interesting.
Yeah, I mean, trying to figure out more about the heart detail would be very interesting for the project, because in some of the data, it shows that when you look at the actual transcriptomic genetic elements, that genes related to the extracellular matrix are downregulated quite significantly. Now, the extracellular matrix is very important for tuberculation to form, because if you don't have the ECM, you don't have the certain processes that are necessary to get these tubercula, which are essentially just say what they are. They're like protrusions that go into the ventricles. And people think they've got quite an important role in actual beating of the heart. And so if you have less tuberculation, are we affecting the actual function of the heart? Our zebrafish kind of suggests that it does affect that. And what's also quite important when you go back to the sort of neurodevelopmental aspects that Oskar suggested, is that you need a lot of blood flow from the heart for the brain to develop properly. So if you don't have these tubercula that are not allowing the heart to function properly, you could end up with brain defects, because it's been shown by other studies that if you have, let's say, a dysfunction in the ventricles in humans, you end up with smaller heads and smaller brains in the babies that have these defects. So having a bit more of a look into how the heart structure could actually also relate to any sort of subtle yet important brain abnormalities, I think would be quite an interesting aspect to see as well. And of course, with the zebrafish, it's quite easy to do behavioral assays as well. So we can actually see, are these genes affecting, say, the anxiety of the zebrafish, the movement, the curiosity, that sort of aspect in the zebrafish.
So that's the one aspect. I think that's more the cardiovascular aspect of it and the developmental aspect. The other aspect that I think fascinates our group. So we can say Daniel's previous boss, Lars, is very fascinated by this aspect. We are very fascinated by the other aspect, which is the working model that's presented in the final figure of the preprint. And this is we delineate a very significant hypothesis, which is that you have a direct crosstalk where one pathway, the non-canonical TGF-beta pathway signaling through TAC1, will always be switched on or off depending on another pathway called hexshot signaling, which we know as a de facto cilia pathway. And hexoid signaling is very interesting because it has implications for both heart, brain development, so on and so forth. We think that many of the ciliary pathways do crosstalk, but we haven't actually ever really delved very much into it inside the field. So the fact that we have this direct link that we really can quite readily prove if we investigate a bit into PKA, we should be able to at least see some kind of effects of it. That is really the signaling aspect where the project would develop further. And I think these two are quite separate because it's kind of hard to do that in the zebrafish, and it's kind of hard to do the vascular compartment in a cell model system.
Yeah, I think they're separate, but if you were to combine the results from both of those sort of separate independent studies, you could end up with quite a nice potential third project where you try and look at, say, drug targets or trying to rescue the phenotypes that we would see. So if we were to be able to understand, say, the cellular mechanisms that Oskar's talking about, do we know of a drug that would be able to target those pathways to be able to try and get a more normal heart in the zebrafish? And that could then be used in the future because we've got human patients who have these syndromes that when you mutate the TAP1 or TAP2 genes that get quite severe clinical symptoms. So in the future, I think combining the cellular and more taking the cellular data and putting it back into the zebrafish with more drug screening assays or gene therapy would be an interesting avenue, especially when you've got patients who are quite severely affected by these genes.
It's actually quite fascinating because it's a really good point, because it corroborates also the human patient data where we looked at some of the, I think it's from a whole exome database that we elucidated. And the fact is that all of these proteins that are involved in this signaling crosstalk really seem to give a very similar syndromic phenotype. So if we can somehow connect that in a zebrafish model, for example, we would really have a very powerful tool to actually understand this. And that's, Daniel, I've talked about this rigorously, I can inform you.
We're quite interesting at the pub. We can clear the room quickly.
And whatever you do in the future, you two should continue to collaborate. You seem to have a perfect working dynamics.
Oh, we're trying to. Yeah.
That I envy. It's perfect.
So Daniel, I know you had you presented a 3-minute presentation on this work for SciComm Connect, and now you're both on this spotLights podcast. Is it right to say you both find like science communication important?
I think definitely, yeah, because of course we can talk to each other, me and Oskar, all we like in the pub and completely bore everybody around us. But if we don't actually tell people about what we're doing, then quite frank, what is the actual point in actually, what's the point in doing it?
Yeah.
So I think it's trying to communicate to not just to other scientists, but to people who are just interested or like the gene mutation may affect them, I think is very important. So I think it's definitely something that we should consider more in not just our lab groups, but in the field, which I think the cilia field's actually doing quite well in doing.
The cilia field is doing a lot towards it. I think it really brings a good point that one of the great paradoxes of all the academia and university systems is that a lot of the funding for public universities, for example, such as the University of Copenhagen, is from regular taxpayers who don't understand what it is they're actually paying for.
Yeah.
And at the same time, a lot of the stuff that we end up publishing gets caught behind paywalls left, right, and center that we only have access to through the university or our workplaces. So I really think science communication is a great tool in actually making people understand and appreciate why this actually matters, because it's not quite, I don't think it would be apparent why this necessarily is as interesting as it is if you don't actually try to communicate it.
And I think some people, like some higher ups, don't even fully understand the concept of science communication, because I'm going to tell a story and I don't know how you're going to edit it in, because so I done the 3-minute SciComm competition, and I let my former boss at the time, not in Copenhagen, in a different university, when I started a postdoc, that I was going to say I was going to miss a meeting. I'm doing this competition, this workshop to improve science communication. Said it was fine. Turned out it wasn't fine. And his idea was that it wasn't important enough to go on this course and to miss lab work and meetings. But of course, that was never actually communicated to me. He said it was actually, yeah, it's fine. And then that resulted in a few other things that happened at this place. I'm not going to name it, but yeah, so they just didn't really seem to understand how science communication is actually really important for the future. And I think they've got quite an imperialistic view on that, which actually resulted in me now actually going to a different lab to start working. So I'm going to be starting a position in Sheffield next month, or this month, actually. No.
That's actually a good gauge of, whether fit into a lab, whether they support science communication and things that you do outside of the lab. All of that is important for your own well-being and for the science too.
Exactly. And hopefully Oskar and I can continue to work together in my new place because
you never know what might come
I'm sure we'll probably end up doing it. I'm going to be looking at the beginning sort of bone disorders in zebrafish, particularly one looking at vascular Ehlers-Danlos syndrome. And hopefully we'll also move on to doing some research into neurodevelopment as well, which is what Oskar is starting to mainly work on now. So you never know, there's opportunities.
No.
Don't tell anyone though. No one knows.
No one knows that yet.
Amazing. Great.
That's it for this episode of Spotlights. We'll see you next time. All our episodes are available on the preLights website, and to keep up to date on each episode's release, follow us on X, Mastodon, or Facebook, or register on our website. See you then.